Hello, and welcome to this webinar titled Control Strategy and Real-Time Release Testing from a Development Line to Commercial Manufacturing, hosted by BioPharma Asia magazine and presented by Tom van der Kerkhof, scientist at Johnson & Johnson. This presentation will be followed by an industry perspective and presented by Sebastian Sowinski, Key Account Manager at Sentronic. My name is Marcus, and I will be the moderator. Before we begin, I would like to inform our viewers that there will be a live Q&A session after both presentations near the end of the webinar. Audience members can send their questions at any time during the webinar via the Questions tab, located directly below your webinar screen, and we will go through these at the end of the webinar. Now, please allow me to introduce our first presenter, Tom van der Kerkhof. Tom completed his PhD in the field of IR and Raman spectroscopy and started at J&J in 2003 with the introduction of NIR spectroscopy in the analytical lab. Initially, the NIR model development was focused on identification of raw materials and models to verify the correct randomization of double-blind clinical studies. Gradually, NIR quantitative models were developed for replacing the more time-consuming LC and KF analysis in a QC lab and using it as a PAT tool. Tom's current task is to optimize the lifecycle management strategy of NIR calibration models to successfully implement them in a QC lab and manufacturing areas and to support regulatory submissions. Furthermore, he's responsible to perform investigations towards new applications of the NIR technique, as well as the use of other optical spectroscopy techniques to increase the QC lab efficiency or to be used to support the manufacturing process. I will now be handing over to our first presenter. Welcome, Tom van der Kerkhof.
Okay, thank you, Marcus, for this introduction. So during this webinar, we will talk about the PAT applications that we use to determine the uniformity of the final blend and of core tablets. Next to the description of the PAT applications, we will talk about the control strategy that we use and a control strategy for the commercial manufacturing area, as well as for commercial products, as for new products that we introduced. And as a last point, we will talk a little bit about of the release of the products that we have produced via continuous manufacturing. So let me start with a little introduction. When we look at continuous manufacturing, it means that we are going to bring in all the individual components as a powder form, and then we apply in a continuous train all the different unit operations in order to obtain our core or coated tablets. The most simple continuous manufacturing just consists of 3 steps, which is feeding, blending, and compression. And on those, I want to stress in this presentation. During the continuous manufacturing, we have 2 types of control tools that we can use in order to prevent that the tablets will be part of the final batch itself. These 2 control tools are residence time distribution, which is based on the mass balance of the feeders, and our process analytical technologies. Then, of course, when we have all the tablets of the final batch, then we have to do batch release. We have to make a decision. Do the tablets that we have produced, do they confirm our specification? If the answer is yes, we can send them to the market itself. So we divide it into 2 parts. So control strategy is assuring a consistent quality of the product itself according to our quality target product profile. And based on our PAT and RTD, we can say that if we have insufficient quality of the products, we can send it to the waste. Actually, the PAT and the RTD that we use, you can see it as an in-process control test. On the other hand, you have release testing. It's testing to release the product to the market itself. Traditionally, you would take a grab sample and you would do lab analysis. This is what we call end product release testing or finished product release testing. But you could also use all the PAT data that has been generated during your continuous manufacturing process. And if we use the PAT data, we talk about real-time release testing. So if we look at the situation that we have within Johnson & Johnson, then we have actually three different types of continuous manufacturing lines. We have, first of all, we have a direct compression line, which is located in our facilities in Gurabo, Puerto Rico. This direct compression line was introduced for a commercial product. So what we have done is we have converted a traditional batch product, a product which was already on the market. We have converted it from batch to continuous manufacturing. The same thing that we have done in our facilities in Italy, in Latina, where also a commercial product we used to convert to a continuous manufacturing process. And then we have what we call a multi-purpose line in the Beersen facilities in Belgium and an identical line in the Latina facilities of Italy. These are the lines that we use to introduce new products in it. Just for information, these I also put the rate of product that can be manufactured per hour in it. Now, the two first ones, where we convert the batch to commercial product, are slightly different than the multi-purpose one. If we look at the last part of our continuous manufacturing, where we have blending and compression, then what we use here in the two commercial lines, that we use the PAT BU tool, we use it before the powder reaches to the compression, which allows us to divert the powder material before it gets to the tablet press. Then, after compression, we have a PAT CU tool, which allows us to analyze the core tablets that have been compressed, and then we can decide to send these core tablets to the waste, or to keep them and later to release them to the patient. If you look at the multi-purpose line, we do it slightly different. The PAT BU tool is there incorporated in the feed frame of the tablet press. At the end of the tablet press, we have also a PAT CU tool, and this allows us to divert the tablets before they are going to the market. And in both cases, we are using the PAT CU tool in order to perform what we call real-time release testing. If you look at the commercial product line, this is the example that we have of the direct compression line in Gurabo. What you see here is how do we tackle our PAT control strategy. Marked in the red circle, you have after the blender, you have the PAT measuring station and a diverter valve. So what we look here as control strategy is to make sure that non-conforming blend must be sent to the waste. How do we achieve this PAT control strategy? Is by first, we have to do experiments. So we have to know what is the time that we have between the time that we measure the product and the time that the product reaches the diverter valve. In this particular example, where we produce at 40 kg per hour, we have 35 seconds difference between the PAT measurement and the time at the diverter valve. Our PAT data recording, what we do is we measure one spectrum every 8 seconds. Then we are going to do a data evaluation, so every 8 seconds, and we do it at 3 levels. First, we look at the spectral quality test, which is in this case an Hotelling T-squared value. Second, we look at the moving block average of 3 consecutive measurements, and we look at the range that we have during the moving block. If one of those 3 fail, then we have to switch the diverter valve into the reject position. This is done automatically when a fail is observed. Before we can switch the diverter valve back into the accept position, therefore also we have to fulfill 3 requirements. First of all, of course, needs to be that the BU acceptance criteria must be met. Second of all, we are going to check our feeding system rates, and we are going to wait for a blend homogenization time. Just to put some figure around it, we have seen in the past that sometimes we had to divert a little bit, and diversion circle means that you would divert approximately 3 kgs of material. So this is done for our commercial product. Now, let's take a look at the new product direct compression line, which is very similar. You again see the four different feeding stations. You go to a blender, you have a second feeding station where you add the lubricant, you go to a second blender, and from the second blender, you go to the tablet press. At the end of the tablet press, we have 3 different ways that the tablet can go. The tablet can go and be collected into what we call a temporary tablet collection drum. That's one way. The other way is that the tablet can be sent immediately to the waste. And the third way is that the tablet can go to our PAT-CU tool. Now, process control here means that we have to eliminate non-conforming material from the final batch. So our first way, divert location that we have, is bringing the tablets at the end of the tablet press to the waste. In order to do that, we use 2 different tools. We use the residence time distribution model, and we use the PAT-BU tool, where we measure the API concentration in the feed frame of the tablet press. Then a second diversion tool is that we can remove a temporary tablet collection drum. For example, if our PAT-CU tool tells us that the tablets in that particular drum are not conforming, then we can still remove that drum. If the drums are not removed, then the combination of all the temporary tablet collection drums will make of the final batch. And that final batch, of course, needs to confirm the specification, and that is done not via end product release testing, but via real-time release testing. In other words, we take into account the PAT data that we have collected during the manufacturing process itself. Okay, what are the PAT tools that we have in place? As mentioned, we have a PAT-BU tool. For this PAT-BU tool, we use a Sentronic device, where we do the reflection measurements on the final blend powder. And what you can see here in these pictures is a photo of the probe itself. This is the left bottom picture. Above, you have a schematic drawing how it's built into the feed frame itself. You can see that the powder comes in and we measure at the top of the feed frame. So the red dot is the NIR sensor that we have. If you would take now into account the measurement area of the probe itself and the material that comes into the feed frame, by doing some simple calculations, we can estimate that by the Sentronic probe itself, we do measure approximately 13% of the entire batch that we produce of it. In comparison, we have the second tool, which is the PAT-CU tool, which is based on a Fourier transform NIR principle, where we do transmission measurements on the core tablets. That is more or less equivalent to our lab NIR spectrometers. If you would calculate there the sample volume that we have measured as compared to the entire batch that we produce, you can see it's of course depending on the manufacturing speed, etc. But it's approximately in the range of 0.01%, or in other words, it's approximately 1 tablet of every 10,000 tablets that have been manufactured. So you can see that with the BU tool, we can actually measure a much higher amount of material. Now, let's talk about the control strategy. When we are developing our new product to be manufactured on a continuous manufacturing line, we want to go to a staggered implementation of the control strategy, because we know that the PAT tools or the PAT applications, it takes some time to develop, and maybe they can also not be developed for every product itself. So it depends on the applicability and the readiness of the PAT tool. How do we see it like this? So I've put here a table where you can see the different stages. The stages represent kind of the life cycle of your product development. And then you have the 2 diversion locations, 1 and 2. 1 at the end of the tablet press, 2 is the complete temporary collection drum itself. If we look at stage 1, which we will use for our clinical batches that are going to produce, then we use only for diversion location 1, we will use the residence time distribution process model. For diversion location number 2, we just do some sampling, manual sampling, send the tablets to the lab and analyze them in the lab with LC technique. Then as the product evolves, then we estimate that generally we will have the PAT-CU tool a little bit earlier developed and validated before the PAT-BU tool. So what the second stage that we apply of a control strategy is that we use the RTD process and we use the PAT-CU tool. We envision that we can use this for clinical and primary stability. In the most ideal case, we will have the PAT-BU tool and the PAT-CU tool at the same time validated so that they can be used. And then we come into control strategy number 3, which is the preferred control strategy that we want. And this is the control strategy that we want to use for our validation batches and for our commercial batches. As noticed here we have also a fourth control strategy stage where we don't use the RTD process model. It can be that when you are in the commercial process that there will be slight changes to the process which impact your residence time distribution model, but it does not impact your PAT-BU model. In that case, we still want to continue in using stage 4. But in general, what we strive to as a company is to use stage 3 as a control strategy. Now, let's take a look at our control strategy. Our control strategy just means prevent that the final batch contains non-conforming core tablets. As mentioned, there are 2 diversion locations. First of all, prevent that those core tablets do not come into the temporary collection drum. We use the RTD and the Sentronic for this. And second of all is prevent that a collection drum with non-conforming tablets will be part of our final batch and can be released to the patient. And therefore we use the PAT-CU tool itself. Now let's take a look at the 3 different tools. First is the prevent of the non-conforming tablets to be part of the collection drum itself, diversion location number 1. We use a residence time distribution process model. This allows us to predict based on the mass balance of the feeders what is the API concentration at the core tablet level. How do we say now, how can we reject certain tablets? Is what you can see here is maybe on the screen is that we have a red line indicated which is the specification limit. Of course, when the prediction of the API concentration is above the specification limit, then of course we need to reject. When the prediction is below, then we can accept the core tablets again. But you have to think that our RTD process model has certain uncertainties. It is based on experiments itself, so you have material variability, you have process parameters variation, you have replicates, you have analytical and procedural variability. So instead of putting the rejection limit at the specification limit, we built in a certain uncertainty, which is the yellow line on the graph below. So we already start rejecting the tablets once we are above this internal uncertainty limit, and we only start accepting the tablets again when we are below that one, as you can see in the picture below. That's the first tool. Then the second tool is the PAT-BU analysis, where we use the Sentronic probe for. If we use a PAT-BU tool, what we try to do with this NIR spectroscopy is always we try to develop a quantitative model based on a PLS algorithm. So we want to predict what the drug substance concentration is or the API concentration. On the other hand, for certain products this is not possible. We also can do an indirect determination where we use a PCA analysis or a PLSDA analysis itself. So what can we measure with this Sentronic tool? We can of course measure manufacturing deviations. If there are deviations in the feeders, in the mass balance of the feeders, we see it with our Sentronic tool. Of course, we also see it with the residence time distribution model. But the Sentronic tool has the advantage that we also can see special causes. There can be material buildup and certain release of material buildup. This is something that you are not able to see with your RTD process model. You can only see it with the PAT-BU tool. So actually, our PAT-BU tool can do a bit more than the RTD tool. So in practice, what will we do is we will continuously measure, and in this case, we will measure every second is a new measurement, and we get results for the drug substance concentration in the blend. At a certain moment, this drug substance concentration goes above the limit, and immediately we will start diverting. From the moment that it drops below the limit, then we don't stop diverting, because there is still some time between the PAT measurement and the outlet of the tablet press itself, which means based on RTD knowledge, we are going to define what this time is. So we take a certain buffer period, as you can see in the graph, in order to keep rejecting the core tablets. Then the third tool, as mentioned, is the PAT-CU analysis. Therefore, it's important for us to make a direct link between the CU analysis and the collection drums itself. Therefore, we use time tags. So each time tablets go to the PAT-CU tool, we receive a time tag. And then on the other hand, we have the time tags when the temporary collection drums are changed. So we can always link the measurements of the tablets to the correct collection drum itself. Of course, here there is still an open question. What would be the required sampling frequency that we need to achieve to control our process? And we will come deeper in this topic in the next slides. So, the sampling frequency itself. What should we take into account? Where are we in our product? Are we in development, clinical or commercial product? What is the actual throughput? What is the sample size that we want to achieve? What is the level of representativeness of the batch? What is the applied control strategy, etc.? And what is the risk assessment? And risk assessment is always performed by taking into account what is the risk if there is a deviation and we are not picking this up in the final batch. Now, let's go through some different scenarios. So the first scenario of the control strategy is actually our preferred one, which was in one of the previous slides. It was stated at stage number 3. So we have all three tools available. We have the RTD tool, we have the Sentronic tool, and we have the PAT-CU tool. The benefit of this scenario is that with your Sentronic tool and your RTD tool, you actually already measure all the potential deviations that can happen, manufacturing deviation and special cause deviations. So in other words, the CU tool is only used to confirm what already your PAT-BU tool is saying and what your RTD tells you. So the sampling frequency that we have to apply can be rather low. It's more a sampling frequency in order to use this data for a real-time release. Second scenario that is possible, that is what we called in the previous slides as stage 4, is that we have the PAT-BU tool available but not the RTD process. Then we are actually in the same situation as scenario 1, because the PAT-BU tool allows you to see any manufacturing deviation and special causes deviations. So in other words, the PAT-CU tool is again a confirmation of what the PAT-BU tool already tells you, and we can do a rather low sampling frequency. If we come into the next scenario, is scenario number 3, which coincides with stage 2 of the previous slides. There, the situation is that due to the product, we were not able to build a PAT-BU tool. This means that at the tablet press itself, we can only remove the tablets that were due to a manufacturing deviation, but not if there was a special cause of material buildup or something else. We cannot remove these tablets at the end of the tablet press. So there is a certain risk. We can still use the PAT-CU tool to confirm the RTD, but we also need to do a risk assessment. And during that risk assessment, during development, we need to define what is the causes. And special causes can be material buildup and release itself. You can assess in your risk assessment that this is low, then you could still use the same sampling frequency as scenario 1 and 2. Let's switch to the fourth scenario, which becomes a little bit more different. In the case that we don't have any control tools, that we can already remove tablets at the end of the tablet press, then it's very important. Then your PAT-CU tool is actually your in-process control tool. It has to replace the RTD and the PAT-BU. So that means that you have to do actually a much higher frequency of sampling, because you need to see what are the manufacturing deviations need to be captured by your CU tool. Only if you can do a risk assessment, then there might still be a chance that you can do a lower sampling frequency. But in general, to control the process, you need here a higher sampling frequency. And then there is, of course, scenario 5, which is the worst case scenario. And that is the scenario where none of the PAT tools are capable to perform, and also no RTD process can be achieved. In that case, again, you have to do risk assessment, and you will need to do manual sampling and lab analysis itself. So these are the different stages and scenarios that we can have with the control strategy. Our preference is, of course, stage 1, where we have RTD process, we have Sentronic BU to divert tablets at the end of the tablet press, and we have a PAT-CU tool in order to confirm it to confirm your BU and your RTD. Then, let's take a look at release testing. What you can see in this table here is at release testing for most products, the tests that you need to do are appearance, identification, assay, purity, content uniformity, dissolution, microbiological purity. Traditional end product testing is then, of course, that if you look at identification and assay and purity and content uniformity, they are generally done by HPLC. We already know that NIR can also be used in the lab to perform identification, assay, and content uniformity, of course not for purity. And then dissolution can be performed via dissolution method or via a surrogate model where you base yourself on the NIR spectra that have been collected. That is, of course, a traditional finished product testing where you take a grab sample at the end of your batch process. If you look now to the continuous manufacturing line, we can do real-time release sampling. And that real-time release sampling can be at the continuous manufacturing line. And for these different tests that we need to do, we can use the PAT-CU tool itself. And we can use for dissolution a surrogate model. If any circumstances the PAT-CU tool is not in use or cannot be used, then you can still do real-time release testing, but you have to think that you still need to do the NIR offline in the lab. Here, the real-time release testing is done then on the core tablets and not on your finished coated tablets itself. Now, let's take a look at real-time release testing. We have the core tablet manufacturing. As mentioned, the core tablets are manufactured, they go into temporary collection drums. From the temporary collection drums, they might first be coated or not, then they go into the final batch. The final batch, let's visualize it as one big drum. That final batch gets a batch number itself. Then we go to packaging, and the packaged material goes to the patient. When we do a release testing, so every blister or every bottle that we make is always linked to the final batch or lot number, but there is no link available between the blister and the temporary collection drums itself. So, if you would do a release testing, what would you put on your certificate of analysis? we believe this should be representative for the final batch. So then there are two options that you can apply. Either you use one overall value per specification, so for ID, assay, content, uniformity, you put one value, or you can put a list of values. Namely, it's one value per tablet collection drum for every temporary collection drum. As a company, we decided in order to go by one overall value per specification. And of course, the control strategy we have been discussing in the previous slide. If we look now at the real-time release testing, we use the PAT-CU tool. It means that a single result, an NIR spectrum, for each analyzed tablet we have. So we do identification. It's just confirming that the API is present in the tablet. We do assay and content uniformity, which is just determining the API content of the tablet. And we will do dissolution via surrogate testing. Then there are two key questions that we need to answer. Is first of all the number of samples that we need to use for release testing. And we already mentioned that we can use the same data used for the in-process control, so your control strategy and your release and your real-time release testing. So, and we can do this for assay and content uniformity. The second question that we need to do is the acceptance criteria. If you look at the regulatory guidelines, like the United States Pharmacopoeia or the European Pharmacopoeia, we already noticed that for content uniformity and assay, there is a chapter which does an extrapolation for large N. So if you look in the European Pharmacopoeia, there is a separate chapter for which you have done content uniformity for 100 or more tablets of a batch. For dissolution, however, there is not such a chapter available in the Pharmacopoeia. So therefore, what we are doing is we are trying to extend the current Pharmacopoeia to this large N ourselves. And by this, I just want to acknowledge several of my colleagues who have been involved in this work. And I want to give the word back to Marcus.
Thank you, Tom. Now, before I introduce our next presenter, Sebastian, once again, I would like to remind our viewers that there will be a live question and answer session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the Questions tab, located directly below your webinar screen, and we will go through these at the end of the presentation. Now, please allow me to introduce our second presenter, Sebastian Sowinski. Sebastian is the Key Account Manager in the Spectroscopy Business Unit at Sentronic in Dresden. In this role, he is responsible for customers in the pharmaceutical industry and supporting them from the first proof of concept for PAT implementation to the final installation and qualification. He is also the main contact and support for internal partners of Sentronic. Sebastian holds an MBA and joined Sentronic in 2000, starting in Spectroscopy Business Unit and providing PAT solutions to the pharmaceutical industry as early as 2004. He gained a comprehensive knowledge of near-infrared spectroscopy and multivariate data analysis, as well as in all aspects of the application of PAT in different stages of the development and processing of solid dose pharmaceuticals. I will now be handing over to our second presenter. Welcome, Sebastian.
Thank you for the introduction, Michael. Thanks to Tom for the very interesting presentation he gave. Good morning to everybody who joined us today. I'm going to bring over a few more technical details, a few application examples, and features of our used technology, which are an advantage for these applications. First of all, I would like to start with a very short introduction to Sentronic as well as to the business unit process spectroscopy I'm working in. Sentronic was founded in 1993. It was founded in Dresden and is still located here. We are a highly qualified team of about 20 people. With this 20 people, we are working in two business units. One is the optical chemical sensors, where we developed and produce oxygen sensor for dissolved oxygen with main market in fish farming. The second business unit, and that's the one I'm going to talk about today, is optical spectroscopy. And our main product here are processed near-infrared spectrometers with a core market or focus market of pharmaceutical industry. And this, in order to serve this market, we have a quality management in place, which is confirmed to GAM5, and we had to have a couple of audits in the past for our quality system. The two main solutions we offer today are the Sentronic FO, which is shown on the bottom left side, as well as the Sentronic BU, Sentronic DA for monitoring of bin blending processes. The unit operations our systems are used in are the granulation, wet and dry granulation. There, the CPPs, as most of you probably know, are the homogeneity of the material, the binder addition, the granule growth and development, as well as the endpoint of these different processes. Once it was, or in case it was a wet granulation, the drying is most likely the next step, and there again the Sentronic FO can be applied to monitor the moisture in real time and to have the endpoint determination of this drying process. For blending, we can differentiate, or we have to differentiate into batch processing as well, or continuous processing on the other hand. The main process parameter or critical quality attribute here is, of course, uniformity, the content of the active ingredient. In case it is a batch process, the endpoint of the process itself. Last but not least, or the final step within a typical production line is, or the last one we are active, is compression or encapsulation. Again, here the homogeneity is matter of interest, or it's a critical parameter for the product. And the last step shown here is the coating thickness or the coating process, and the CPP is the coating thickness. This is a field where we are not active yet. The two system types are using different technologies. So we have the Sentronic FO, which contains a dietary spectrometer, and you can see on the top left side classification within available spectrometer types technologies used to acquire an NIR spectrum. The Sentronic BU is using a tunable laser MEMS-based spectrometer. So both, or all three of them, and both technologies are full spectrum analyzers and are categorized as monochromators. Let's go a bit more into the details of this NIR-based PET solutions we offer, or in general PET equipment. So on the left-hand side, you can see some very well-known, established general requirements to any PET equipment. So the equipment itself has to conform with USP, EP, in general pharmacopoeia requirements, and it has to be the capability to get validated against these requirements. It doesn't matter if it's a continuous or a batch process. It has to have a very long time, very long-term stability, and also over this long-term high reliability and robustness. The owner of a system, of a PET tool, always is interested to minimize the frequency of required services, and for integration into new but also into existing lines, it's important to have flexible interface option for software and hardware, and to have multiple options for spectral analysis and multiple parameter output based on NVDA. Looking into a continuous manufacturing line, there are a few additional or few more relevant requirements. Very often it is a measurement of a moving product. So we have different speed of powder, granule, or product moving through the line. Measuring directly in line, the measurement system itself has to have or has to be insensitive against any vibrations, any disturbances based on these vibrations. It has to be capable of running 24/7. And here it is even more important to have the fully minimized need of any user or the operator interaction, in the meaning of doing some standardization, external measurements, qualification, validation, calibration, whatsoever. As the system is installed and it isn't that easy to remove the probe and to take it out to do some offline work. Very often we do have a need of a very high acquisition rate in the meaning of seconds as a measurement interval, in the meaning again of value to value, parameter to parameter, and real-time output is needed here in order to raise any alarm or to control a diverter, as shown by Tom. The system we use in continuous lines, or we offer for continuous lines, is the Sentripad FO, which can be combined with different central interfaces or probe interfaces. The Sentripad FO itself is a flexible analyzer unit based on a diode array spectrometer, which is a very fast technology to acquire NIR spectra. It can be applied to a variety of unit operations within solid dose manufacturing, and this can be continuous lines as well as batch lines. And the system itself has an embedded PC, which makes it standalone, which makes it independent from external PC hardware or control hardware. It can be hooked up directly into a local area network, into the production IT environment. And the Sentripad FO has also the possibility to be configured as a multi-channel system, which allows them to monitor different measurement positions, measurement ports within a line. The heart of the Sentripad FO system is a diode array spectrometer. We are using a Hamamatsu G9206 detector. There is a schematic view about the sizes inside of this spectrometer itself. It is an array consisting of 256 pixels, which gives a pixel dispersion of 4.2 nanometer. We are using a slit with a width of 100 micrometer, and the connection of the input fiber is a 400 micrometer fiber. The used spectral range is 1100 to 2100 nanometer, and even with the array as shown goes up to 2200 nanometer. The signal to noise ratio at the last 100 nanometer is too low, and that's the reason why we reduced it down to 2100 nanometer. On the right-hand side, you can see a typical layout of a diode array spectrometer. It is simplified here for this presentation. It shows only the concave grating, and it doesn't show any mirrors or other optical components in the light. The fiber optic input is at a fixed position, and all optical components, the fiber input with slit, the grating itself, and the detector are fixed to the housing and frame, and there are no moving parts for this spectrometer or optical bench, which provides excellent long-term stability and makes it possible to use this system fit for purpose for this continuous line. So it is fully robust against vibrations. It has a very low risk of alignment. If the system itself is moved, it has a superior long-term stability over many months and years. Further advantages of a diode array spectrometer is the fact that all wavelengths are collected at the same time, so it's more like a photo, and there is no noise picked up as the sample presentation changes. During the measurement of a moving product, that can easily happen, and I have later on a few slides which show how this can be handled with the NIR diode array spectrometer. Further, the diode array spectrometer can be used with fiber optical probes with fiber optical cables, which makes it easy to install the spectrometer itself away from the process in the technical area or somewhere away from the line, and the probes are the only part which are interfacing with the process itself with the material. However, diode area spectrometer are sometimes claimed to have not a wavelength stability which is as robust as a FT spectrometer or scanning system is. As this is true, but there are solutions to overcome this issue. And the next challenge we have, as we have with any optical measurement system and with majority of measurement system, is that there are some drift effects over time, which again is a challenge when the process is going for hours, days, or even weeks. The last challenge, sometimes blamed the diode area spectrometer for, is that the resolution is not as high as it is with an FT spectrometer. Well, that's true, but the NIR bands of many analytes within small molecule pharmaceutical or also with food ingredients, other industries are broad enough with diode area spectrometer optical resolution of 12 nanometer. The first 2 points we solve with with internal filters and internal standards. For the wavelength stability of the system over time, we have a Fabry-Perot filter inbuilt into the unit, and this Fabry-Perot filter creates a periodic constructive destructive interference. And this interference characteristic of any individual unit is measured during the factory calibration, during startup of any system, and it represents a known good calibration state. And during use of the system, this internal filter is measured again based on a interval which is configured, and any shifts which may occur due to a shift of the optical module or due to fast changes of ambient temperatures can be corrected based on this internal filter. And this results in a stability as shown on this slide. We have the black curve with the axis on the right-hand side, which is an ambient temperature of about 15 degrees C going up to 30 degrees C. And we have a well-stable wavelength accuracy of the unit. This test was done in a climate chamber. The standard measured was a WC 1920 standard, which is a common standard for system validation for wavelength uncertainty and wavelength reproducibility. In order to overcome drift effects, as said, as you have on every optical measurement system, we have a standard which is inbuilt into the probe, which is used as an active reference. The setup or the sketch here is showing the normal sample measurement. So we have the material measuring continuously, and we have an internal standard which can be measured at any time during the process, and which is used as an active reference. And this gives us 4 different type of spectra. So we have the sample or the external reference position, and we have the correction or active correction and signal at time point 0, which is the external standard measurement, which is the time of referencing the unit, as well as the C term, which is measured during the process. This explained here a bit in more details. And to keep it a bit simpler here, the dye corrections and the probe background corrections, which are also part of the calculation of the spectra, are not included in the formula shown. The result of this is shown here. You can see 2 trends of absorbance value for a continuous measurement. For this experiment, we used to we measured a 99% standard, which is a 100% reflectance, which again is then an absorbance of 0. And the blue curve, which remains around 0, which does not show any significant drift effect, is the output of a central-pad FO in combination with a central probe DRLS, while the red curve is an internal signal which was calculated offline and which does not include any drift correction or active reference effect. And the period shown here is, as you can see, about 12 hours. Let me continue to go into a few more application-specific details. First, I'm going to show you something about blend BU or BU monitoring during a continuous manufacturing. This again shows the CDC process as presented already by Tom. There are two different measurement positions. One is a measurement in just after the outlet of the blender, of the continuous blender module, where the probe is installed in a chute and predicts the powder or measures the powder and granule or granule while it is passing by and while it is transferred to the tablet press. Here we have the challenge to handle a sample presentation which is not the most constant one. Sometimes there is some pulsation in the amount of material due to characteristics of the blender outlet of the transfer into the line. The second challenge we have here is that the time between the material is passing the NIR probe and the position where it can be excluded from the process in case it's out of spec material is a very, very short one, where we have only a few hundred milliseconds to max 1 second to still control a diverter via the NIR spectrum. This time can only be realized with a dielectric spectrometer module, which has typical measurement time for one individual spectrum of around 10 milliseconds. With an averaging applied, it is still in a range of a few hundred milliseconds. And the differences in the sample presentation and the amount of material passing by the probe can be handled by a spectral selection algorithm, which I will show you later in a few slides. While this is a fast-moving product, there are also lines with a relatively slow throughput, and then the 5 millimeter diameter of the central probe DRLS may not be sufficient to have a fast sample or to have the time to measure a unit dose equivalent in a relatively short time. For this installations with the slow throughput, we developed a 7-spot probe with each individual measurement spot having a diameter of 5 millimeters, so the time to unit dose equivalent is reduced by factor 7 for this type of installation. The development here was done together with GEA for one of our main customers, and the probe can be used with the GEA Lighthouse system, where automated reference measurement and the probe cleaning is available as well. The detection can be done for either each individual measurement spot or with an optical merge with one single core fiber. Looking into a CDC, we have another possibility for a measurement, and this is a measurement in a feed frame, which Tom presented. And here the central probe DRLS is used and mounted inside or close to the feed frame. It is the very last position where material can be measured as a granule, and the very last position the material can be characterized prior to the compression itself. It's a very fast measurement with a dielectric-based system here allows the monitoring of homogeneity and potency of a higher amount of material. You got this comparison of numbers, and Tom mentioned, I guess, 13% of the entire batch material is measured in this position. We do not have an impact of the petal surfaces while the petals are moving and moving the product into the die of the press. This sometimes requires a special design of the petals, but I know there are also installations which do not have this special design. And as we already saw, this is one key enabler for real-time release testing, for real-time release strategies in the process. Talking about moving product, we may have the following situation where we have an example spectrum of lactose shown on the middle top. And this spectrum shown there is the average out of 10 spectra which are collected within 100 milliseconds. At the bottom left side, you can see each individual spectrum along with the timestamp, and you can see the total measurement time is 100 milliseconds. And the offset, which is shown here, which is visible here, is due to changes in the sample presentation. It could be just the distance between material and probe, but it could be also the amount of material which is moving by. And if we plot one wavelength here or the offset as a dynamic change within this 100 milliseconds, we can see what happens, or on the last diagram, last spectrum, we can see what happens with a scanning system. So for a scanning system, this dynamic change is only in a certain wavelength range and introduces a new peak, which is obviously not for real, while the dietary spectrometer still has a product spectrum as expected and as measured under static conditions as example spectrum. A second example for applying a dietary-based near-infrared tool, I selected the high-shear wet granulation and fluid bed granulation drying. For high-shear wet granulation, there are three different process phases. The first one is the trimixing phase or trimix phase. The second one is the addition of the binder or just the liquid. And the third and last one is granulation kneading phase. One challenge here is that the material tends to be very sticky while the binder is added. And this has a high risk of window falling. And we solve this by applying internal heating to the sapphire window as a sample interface itself. By this heating, the viscosity of the binder remains on a level where it doesn't tend to stick. And with the heating and in combination with the forces of the moving product, the window remains clean, while everything else is coated inside the granulator bowl. The PCA plot at the left side bottom shows the three different phases. You have a trimix phase, which is very, very fast changing for all three batches measured and shown here. Then you have the liquid addition, which shows, or which is shown as an increase in the first component, and then the final granulation feeding phase to the endpoint of each of the three batches can be monitored and traced as well. As most of you probably know, is that NIR spectra are affected by physical as well as chemical changes. So NIR can even be used here to have a trend of physical changes, of granule growth, as these changes during this unit operation. The last application example I have here is the monitoring of the fluid bed drying or also granulation. It's again a challenging sample presentation. The probes are installed inside the process, slightly above the bed before it is fluidized, and once it is fluidized and comes up, spectra are acquired. And for a drying, so obvious CQA here is final moisture, and this can be trended on either a PLS model based on LED reference values, or it can be trended on a PCA as shown here. As already mentioned a couple of times, we do have challenging sample presentations in a high shear weight granulation inside a fluid bed system, but also when measuring material on a passing by a probe in a chute. And this leads to an offset of the spectra due to different amount of material, which shows a very varying spectra characteristic in the meaning of an offset. If I now watch to one wavelength selected here, I have a trend of the absorbance value at this wavelength over time as shown here. Each blue dot represents one individual measurement and represents about 10 milliseconds of process time. And if I then apply an acceptance band where I say, well, this wavelength has to be within this acceptance band, and then I make a good bed decision as shown in pink, I only use these which are considered as being good based on the spectral properties at this one wavelength. I get a set of filtered spectra which is narrowed down here and which removed all of these variations which are due to sample presentation changes in sample presentation, which then is a set of spectra I can easily use for a PLS, for PCA, for any MVDA methods used to generate information related to the process based on the NIR spectra as data. With this last slide, I would like to thank everybody for joining us today, and I'm open for questions, comments, remarks, and I would like to hand it back to Markus.
Thank you, Sebastian. So now we will begin the question and answer segment of the webinar. Once again, I would like to remind our audience that you can still send your questions via the questions tab located directly below your webinar screen. And so we will begin now then with our first question. Can the PAT-BU tool in Sentronic be used for real-time release testing? Tom, would you like to take this question?
Yes, Marcus, thank you. Yes, the PAT-BU tool can also be used for real-time release testing, but it is a little bit more difficult. If you look at assay and content uniformity, normally assay and content uniformity are calculated based on the API content, meaning that the API content of a tablet is if the label claim tells you it should contain 25 milligram, then you have to put it, the content is 25 milligram. While with the NIR measurement itself, you don't measure the content, you measure the concentration. So the API concentration as a percentage weight over weight, more or less. So in order to use the PAT-BU tool also for assay content uniformity, I think you can use the NIR data, but you have to combine it with the tablet weight data that you achieve during your process. And then a combination of both of them can reveal you what the API content is, and then in that case you can use the PAT-BU tool as also a tool for real-time release testing.
Thank you. So for our next question, the wavelength accuracy for NIR systems is specified in USP 1119 and EP 2.2.40 with an allowed deviation of plus minus 1n, which means that two systems can have a system of almost 2 nm. Has Sentronic an approach to tighten this as a pharmacopoeia limits can be a problem for the PLS model, and especially for model transfer? Sebastian, would you like to take this question?
Yes, sure. Well, that's fully correct that the limits which are defined by pharmacopoeia documents are a challenge when it comes to a model transfer. And we have been pushed very hard by a customer a couple of years ago to tighten these ranges. And we developed a very dedicated calibration process for the dietary spectrometers in combination with the shown Fabry-Perot filter module to tighten this down, and we are much lower than USP or EP criteria. We are typically in a range of about 0.5 nanometer when measuring an external standard, and with a unit-to-unit comparison, which is even lower, and also the long-term stability based on ambient effects like the temperature, which I have shown on one of the slides, is narrowed down to 0.1 nanometer. So overall, yes, these units are better with respect to wavelength uncertainties as defined and required by pharmacopoeia documents.
Thank you very much. For our next question, how can you define the time for rejecting tablets based on PAT-BU tools? Tom, would you like to take this question? Would you like me to repeat for you?
I'm sorry, Marcus, my
No
phone
problem.
was on mute.
No problem.
My apologies.
Would you like me to repeat the question, or did you hear it?
No, I did hear the question.
Okay.
It's about the time that you divert
That's
the
right.
tablets based on the PAT-BU. My apologies.
No problem.
This is actually defined indeed by residence time distribution experiments that you do during your development. The most critical point is to find out what is the time difference between the PAT-BU measurement and the outlet of the tablet press itself. What we do is we define this time, so that means that we do the following is, from the moment that the PAT-BU measures a tablet a sample that is non-conforming, we will immediately switch the valve to the divert position, so we don't take into account this time. And then from the moment that it starts, that the BU result is again in spec, we keep the valve in the divert position and we add a certain time. It is the time that we know from our RTD experiments to it. For example, at the 40 kg per hour, it is 11 seconds. We will add those 11 seconds, but those 11 seconds, we will also put some uncertainty on it. So this is still under internal discussion how much the uncertainty is, but you can imagine that we would put something like 20 additional seconds at this particular manufacturing rate.
Thank you very much. For our next question, what is the advantage of the 7-spot probe over the single-spot probe, and when should which probe be used? Sebastian, would you like to say this question?
Yeah, of course. The central probe DRLS, which was also shown by Tom, which is used in speed frame, has a measurement spot with a diameter of about 5 millimeter. So it is intended to be used on a fast-moving product as we have inside the feed frame, or as we have on a chute when powder is passing above the probe window. And it actually defines the sample volume when taking into account the measurements for diameter, as well as the speed of the moving product. If there is a very slowly moving product, we need to increase the sample volume, and the only option here is to increase the measurement spot size, and this we have done with the 7-spot probe, where we have then 7 times individual measurement spot with the same diameter as with for the central probe DRLS, and thus the time to measure a unit dose equivalent is significantly reduced. And as shown in one of the slides, or in the slides related to this MS7 probe, it is most often used with the GEA Lighthouse installation in the PDB section. And from our point of view, our recommendation is to go with the MS7 or the 7-spot probe whenever the material is moving slowly, for example, due to a low throughput line, or through to the setup and measurement precision, for example, in powder dosing valve section.
Okay, thank you very much. For our next question, how do you build your RTD model with color tracer, step change, API spiking? Tom, would you like to take this question?
Yes, and it can be a very short answer. We do step change. So we change the concentration of the API at the end of a run, where we either go above or below, and this is a major benefit that you can also use then that data in order to build your PAT models if you use step change.
Thank you very much. We have a comment and a question at the same time, so I'll read it for you. Thank you, Tom and Sebastian, for the very up-to-date and clear presentation. Great work of the team. And then for your question, during the validation stage of CM, do you perform startup and shutdown 3 times or only perform only once? The batch size is predetermined by API size, batch size or volume, RTD. No, is the batch size predetermined by API, batch size or volume, RTD? Sebastian, would you like to take this question?
I guess that's a question which is better addressed to Tom.
Okay. Yeah. There are actually 2 questions in it. It goes about the validation stage. So your validation batches, as we normally do in a batch process, you do 3 validation batches. Actually, what we have defined in our company is also to perform 3 continuous manufacturing runs. Now, of course, it depends the time of the run. What is the length of the run itself? And therefore, we are not going to do the 3 validation batches with exactly the same length of run. Here we are still into discussion with regulatory authorities, but in generally what we like to do is to do 2 rather short runs as validation batches and 1 run which has approximately the time that we would use for commercial batches. Then your second question is, how do you define the batch size? Is it by the API that is used? I think you mean here, do you include the startup and the shutdown of the continuous manufacturing run, or do you just include the state of control? And to be honest on that, there is still some discussion on it, but what we define at this moment up front is, when we run, is this will be the runtime, and we define it as being the state of control.
Thank you very much. Just a quick reminder before we move on to some more questions, that our audience members can still view this webinar and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you're watching this on demand, then please feel free to send your questions over to me at marcus.osei@biopharma-asia.com, and we'll get those answers to you. So moving on to our next question. What is the benefit of PAT BU tool in the feed frame of the tablet press versus a PAT tool before the tablet press? Tom, would you like to take this question?
Yes, and I hope that Sebastian can also link to it. But there are probably several answers on it. What we see is that we use the BU tool into the feed frame itself, is that of course you only have 1 diversion location. It's the end of your tablet press itself. You divert the core tablets. That's one of the things that we see as a benefit. The other thing is, and maybe Sebastian knows, is what is the volume that you can obtain? I don't know. Is it the same volume measurement that you can obtain if you measure up front, or do you say even up front more? So there are several benefits and potential downsides. The benefit of measuring up front is that you can still divert your powder before it goes in the tablet press. If it's in a tablet press, of course, you can only divert the core tablets. Maybe if it's in the tablet press, the diversion time can be more limited as measuring up front of it. That are potential answers, but I don't have a complete idea what is now the best way to work. Sebastian, do you want to comment on that?
Sure, I can comment, and as you already said, there are multiple aspects here. First of all, the point you raised, that's the sample volume. When measuring a fast-moving product upfront the tablet press, I would roughly say we will see almost the same amount of material upfront as well as in the feed frame. So I wouldn't worry here. One very strong argument for measuring inside the feed frame from my point of view is that it is the very last position prior to compression where the blend uniformity can be detected. So there is almost no risk of segregation anymore. Further argument here is for the timing of diverting out of spec material. When the granule or the material is in the feed frame, it will be compressed to a tablet a few milliseconds to seconds later on, and then you have the outlet of the tablet press, which is a fixed time based on the parameter or process settings of the tablet press, while the time measuring upfront is much shorter between measurement position and position where material can be excluded from the stream, which is more challenging and which then also has a risk of excluding material after the tablet press cannot be really fully related to the measurement position upfront anymore, as there is some hold up due to material collected in the filling section of the tablet press. So all in all, both positions are possible. From my point of view, the measurement inside the feed frame is a better one for timing exclusion reasons, as it is the very last, the very final position. These are the main drivers from my point of view.
Thank you very much. For our next question, do you take into account the BU2 data every second to trigger the diversion, or is it some sort of average between consecutive measurements? How is this influenced by the state of control required? Sebastian, would you like to take this question?
Could you repeat this question, please?
Of course. Do you take into account the BU2 data every second to trigger the diversion, or is it some sort of average between consecutive measurements? How is this influenced by the state of control required?
Well, the final control strategy is not in our responsibility, but I know that for this measurement upfront to tablet press, there are two parallel running MVDA threads. One is taking into account each individual measurement for each second data are required, and the second one running in parallel takes into account a moving clock of sometimes between 3 and 10 predictions, which then obviously has a longer decision time or time to decision. And both streams or both threads can control the diverter with different limits, and material has to be in spec for both threads in order to keep material inside the production line.
Okay, thank you very much. Our next question here. What is the limit of detection for an API using the NIR VESP? What is the lowest concentration of API that can be detected? Sebastian, would you like to take this one?
Sure. Actually, there is no hard value in the meaning of limit of detection for any near-infrared measurement. As a rule of thumb, and not more, I would say something around 5% can be easily measured, quantified with a very low error of the method. But there are some materials, like a very good example here is magnesium stearate, which can be even detected and quantified in a concentration range around 1%. So with a very low. And bottom line here is it depends on the overall mixture, which ingredients are in, as well as the spectral properties of all ingredients, but most importantly of the active ingredient or the ingredient which should be quantified. And this altogether then gives the limit of detection. And in daily life or in daily routine, it has to be tested, it has to be determined with offline measurements, with feasibility studies, with proof of concept studies.
Okay, thank you very much. So thank you, audience members, for your questions. Before I finish the webinar, I would like to ask our presenters, Tom and Sebastian, if they have any closing remarks.
Hi, Marcus. Tom here. I just want to thank you for organizing this webinar. It was very good. I learned some new things from Sebastian. I'm happy with that. And also want to thank the audience for several questions which are interesting. As you noticed during my presentation, we also don't have all the answers. We are also still trying to see what is the best way to continue here.
Thank you.
Yeah, I also would like to thank to Michael and the team at BioPharma Asia for organizing this event or this webinar this morning. I would like to thank Tom for his presentation and all people who joined us this morning. And as it gets more and more important these days, I would like to wish everybody the best, stay safe and stay healthy.
Thank you. And I'd like to take this opportunity to thank our presenters. Once again, that was Tom van der Kerkhof, scientist at Johnson & Johnson, and Sebastian Sowinski, key accounts manager at Sentronic. Thank you for sharing your knowledge with us, and thank you, everyone. Have a great day.